Okay, so here we are, part 2 of the nervous system. Briefly, quickly in review, we've got 6 total types of neuroglia, and we have our neurons. So our neurons are going to respond to stimuli by changing membrane potential, and that's what the focus of this lecture will be on. And then we have our 6 types of neuroglia, 4 in the central nervous system, 2 in the peripheral nervous system. So just kind of keep them in mind as we work through, as they are just our supporting cells in our nervous tissue. By the end of this lecture, you will be ready to take that nervous tissue quiz, which is due on the 30th of November. The only assignment for this section of the course due in November. Everything else is due well after Thanksgiving into December. So 3 functions of neurons that we keep in mind as we get ready to talk about principles of electricity and then talk about the actual action potential and how that works. So neurons are highly irritable. They're going to respond to an adequate stimuli by generating an action potential, which we call a nerve impulse. So in the last section when we talked about an action potential or a nerve impulse travels along the axon of a motor neuron arriving at the neuromuscular junction, we're focusing on the neuron side. And then impulses are always the same regarding of the stimulus. And this is our all or none phenomenon, right? So if depolarization, which we'll talk about in a little bit, but if depolarization hits the threshold, then the action potential is produced. If it doesn't hit the threshold, then the action potential is not produced. So all or none. It's either hit or it's not. So these are your 3 basic principles of electricity. We just briefly touch on them, nothing crazy to add here except for what it already shows. Opposite charges attract. Energy is required to separate opposite charges across a membrane. And energy is liberated or freed or used for work when opposite charges come together. We call that potential energy. Okay, so when it comes together, we have the potential for work to be done. All right, so we have Ohm's law here, and this is the relationship between voltage, current, and resistance. Actually, this term has showed up on many a random crossword puzzle that I've used to keep my brain functioning during this interesting year. If you're watching this in a year after 2020, well, then I didn't re-record this section, and you'll have to reflect on 2020 to think about what I might be talking about. But so Ohm's law deals with relationship between voltage, current, and resistance. In the body, electrical currents reflect ion flow across membranes. Okay? We get 3 pieces of information that come into play. The current is directly proportional to voltage. No net current flow between points of same potential. And the current is inversely related to resistance. So greater voltage equals greater current. Greater resistance equals smaller current. So current is the measure of our potential energy generated by those separated electrical charges. Voltage is the flow of electrical charge from one point to another. That's work. And resistance is any hindrance to the charge flow from substances through which it passes. High resistance would be an insulator, and low resistance would be a conductor. So general principle here is that current is equal to the voltage over the resistance. So membrane ion channels serve a pretty important role. First, though, proteins serve as membrane ion channels. We talked about that way back in like chapter 3, maybe, somewhere like that. Yeah, kind of the beginning of chapter 3. So your Proteins serve as membrane ion channels. Flow through gated channels creates electrical current, so the voltage changes across the membrane. Right, so when you see that symbol V equals I times R, you're talking about the voltage is equal to the current times the resistance. Okay? We have two different types. We have leakage, which are non-gated channels. They're kind of just always open, so no gate, walk through, right? Your gated channels, when they're open, ions are going to diffuse quickly from high to low down the electrochemical gradient. If you see I left a little blank here, flow creates electrical current, so voltage changes. Already said that once, but just in case I left it for you to fill in. We have three different types of gated channels. We have those that are chemically gated. Those open with the binding of a specific neurotransmitter, such as the acetylcholine we saw when we were talking about a muscular contraction, or the process for contraction. We have voltage-gated channels, which open and close in response to changes in the membrane potential. And then we have mechanically gated channels that open and close in response to physical deformation or deformation of the receptors. Okay? So when we're looking at this, ACH was an important one, right, with our chemical gates, right, the neurotransmitter. Voltage gate might be like a sodium channel, okay, a voltage-gated sodium channel, which you might see in a little bit. And then mechanically gated would be something like a sensory receptor for touch and pressure. So those are the differences there with your gated channels. And we talked about chemically gated already once. We'll talk about it again, and we'll talk about some voltage gated later in this section. So changes are the signals used to receive, integrate, and send information. We have two different types of changing membrane potential, and toward the end of this lecture, we're going to compare these side by side so that you can see them with more clarity. But first up are our graded potentials, which are incoming short distance signals. These can summate to generate action potential. They're going to occur when the stimulus causes gated ion channels to open. Their magnitude is going to vary, again, hence why they're graded based on stimulus strength. And they decrease in magnitude as they travel further, because as the distance increases, the ions flow and diffuse through those leakage channels. Our action potentials are long distance, all or none, and what we consider the nerve impulse. And these always begin at our resting membrane potential, which you can see listed here. That's going to be an important number to get to know because it is where we hang out when the neuron is not firing. So I'm going to go over what all these numbers are, what this diagram is showing you here when we're dealing with membrane potential and the creation of an action potential. So we're going to define the phases of an action potential here. So keep in mind the voltage that we mentioned, right, as our starting point. We always hang out at that -70. You can see that this is a fast process because it shows you that over 5 milliseconds, we're kind of carrying this out. Okay? So let's start at number 1. This is the stimulus. Okay, so we're currently at resting. Okay, 0.5 is on your left there as well as on the right of the thing, but we're at -70 as far as our voltage. And we get a stimulus, right? So a quick note about the stimulus. As your stimulus strength increases, the frequency of the action potential will increase, but your amplitude of all action potentials is the same. So as we're defining what we consider to be the nerve impulse, we'll keep in mind that stimulus strength increases frequency. But we're going to talk about just one cycle. So the goal for an action potential is to reach threshold, which is noted on this as -55, could be -50. As you move more toward 0, sodium permeability increases. Okay, the sodium traveling in is going to exceed the potassium out. This is an example of a positive feedback cycle. We're increasing the signal. At point 2, we've gone from -70 to 0, and this is depolarization. Our movement towards 0, so the inside becomes less negative, and the probability of producing that nerve impulse increases. It doesn't stay there though. So as we hit to about +30, repolarization is going to begin. The potassium is going to exit, the sodium channels are going to close, and we're attempting to restore the electrical condition. We overshoot every time, and you'll see that at spot 4, that we go well beyond resting potential. So hyperpolarization occurs as the gates close slowly. Because they close slowly, we overshoot our target. The inside is more negative, closer to -100, and the probability of the impulse is reduced. We then work to return to the resting state. The sodium and potassium gates are closed, and we get our sodium-potassium pump restoring resting ionic conditions. Okay, so we have this range from +30 or 40 to -100, right? So we're at -70, we're at resting membrane potential. We have this all or none phenomenon. Intracellular situations negative, extracellular is positive. When it starts to move towards 0, we're at depolarization. We hit our action potential, +30, +40, and we are already working to repolarize. We overshoot to -100 and return to our resting state of -. This is our general process. Those are the big steps that are going on there. This is going to be very helpful to you. So I'd watch it a few times, look in the book for the steps, write these steps out multiple times if you need to. Pretty good deal here. I'm going to break this down into some smaller pieces and bring your attention to some things that are going on in each place as we move through these slides. All right, so up first is that resting state. So here you can see our sodium and potassium channels are closed. There's some leakage that goes on of both. They're capable of opening, and you'll see that each sodium channel has 2 gates. So keep that in mind, 2 gates, whereas potassium only has 1. We have what's known as an activation gate, which is going to be closed at the resting state, and what's known as an inactivation gate, which is open at resting. So you can see in that first segment of this picture, the resting, how your inactivation gate is open and the activation gate is closed. This means that it's resting potential, we're at -70, there is the possibility that we could do something. So we're sitting here between number 5 on the left and number 1, kind of hanging out in our resting state, ready to do something, we hope. As we move towards 0, becoming less negative, we're making our way towards the hope that we can hit that threshold of -50, -55. We're becoming less negative, and your central nervous system here is going to determine the stimulus intensity and frequency of the impulse. You'll see that as we hit threshold, that these gates, the sodium channels open, and you get this rush of sodium, which then further depolarizes and sets us into that action potential. We trigger the work, right? Once it hits +30, though, the gate's going to close to stop the movement of sodium. Okay, great. Keep watching. So at threshold, we're at -55. Positive feedback opens all the sodium gates. We get a spike of action potential at +30. Again, that's our voltage. So with repolarization, we're at +30. We're working to reestablish resting membrane potential by getting potassium to leave the cell. Your slow inactivation gate closes. The slow voltage potassium gates open. Potassium exits, and internal negativity is restored. Remember, we overshoot a little bit, so what's going on? We go from +30, we want to land at -70, but something's happening, right? We've got an inactivation gate that stops more immediately with sodium, while the actual activation gate closes slowly. So we overshoot, but why? Well, if you go back to look at the picture on the previous slide, you'll see that sodium has the inactivation gate and potassium does not. We hit that -70, but the gate closes slowly, so we end up overshooting, right? It isn't closed yet, right? So remember, repolarization restores resting electrical conditions of the neuron, but we still have to restore our resting ionic conditions, which is going to be done by the sodium-potassium pump. So while the neuron is insensitive to stimulus and depolarization at this point, we've reset, our resting electrical condition. We've got to finish the resting ionic condition. So your sodium-potassium pump is going to help balance us back out. So during action potential generation with voltage-gated sodium channels open, a neuron cannot respond to another stimulus no matter the strength. We have two things working here. We have our absolute and relative refractory periods. Our absolute refractory period is from the opening of the sodium channel until it's reset. This is going to enforce our one-way transmission and ensure the all or none. It's either going to go or it's not. The relative refractory period follows the absolute. We get our sodium at resting state, some potassium open, right? That's our repolarization. We have an elevated threshold for action potential. We could generate again with a strong stimulus. We could, once we're in that relative refractory period. Okay? So it's absolute, then relative. And it's relative because it depends on the stimulus strength if it could be generated again. So the conduction velocity of the action potential depends on two factors. One is the axon diameter, and the other is the myelin sheath or myelination. With the first one, the larger diameter fiber has less resistance, so therefore a faster impulse. Think of the difference between the amount of water that can flow through a garden hose versus a big fat fire hose. Right? So if it's larger, you get less resistance and faster impulse. With myelination is there to help speed up transmission. So we have fibers that are unmyelinated, where the influx of sodium spreads through, and that spread has an unfocused depolarization, so therefore it's slower. You can see that in the bottom part of that picture that I've included, how it kind of spreads out and it's kind of moving. You can see positive and negative charges on both sides of that unmyelinated axon. And then we have our myelinated axon. This is going to protect that sodium-potassium spread, and the action potential hops at the nodes of Ranvier, right? So action potential can only occur at a node of Ranvier, which is that gap between Schwann cells. And we have this process known as saltatory conduction. This is where the action potential leaps from node to node, which is approximately 30 times faster and more focused. And your voltage-gated sodium channels are at the nodes. This is going to be way faster. So if you look at the picture here, you have these unfocused action traveling, whereas in the myelinated, you've got your arrows showing you that focused spread of depolarization, right? So here's something interesting. With MS, you get a short circuit in the nerve impulse causing demyelination. So with that demyelination, you then get this unfocused depolarization, which causes a change in the conduction velocity for your action potential, which can change that muscular contraction work. Okay? So that's just a quick little tidbit on the side. So synaptic transmission is the part of work being done at the synapse or the synaptic cleft. So the synapse is the connection between neurons or a neuron and an effector. The synaptic cleft itself is the gap between the presynaptic neuron and the postsynaptic cell. Right? So the presynaptic neuron is sending the impulse toward the synapse, and the postsynaptic cell is carrying that impulse away. This is a chemical event. This is chemical transmission. The presynaptic cell releases a chemical, a neurotransmitter, such as ACH, that stimulates the postsynaptic cell. So for example, you get this at the neuromuscular junction. We have the presynaptic motor neuron releasing the ACH to stimulate the sarcolemma on the postsynaptic side of that activity. We have synaptic vesicles that are going to hold the neurotransmitter in the axon terminal of that presynaptic neuron. And then you have a receptor region on the receiving side to respond to the chemical neurotransmitter. Okay? There are a number of steps, okay, and it's in the picture, but I'm going to walk you through them. You can find this picture in the book, I believe, as well. Let me quick flip a page or two and see if I can tell you what page it's on as we get going. So I have like six steps that we're going to look at here for the chemical synapse transmission. And you can find it on page 415 in your book. Okay, so first we have our action potential that's arriving at the axon terminal. Okay. We're then going to have the depolarization of the presynaptic axon, opening those calcium gates. The entry of calcium is going to cause the release of the neurotransmitter from the synaptic vesicle. In the case of our previous section on muscles, that would have been the ACH. That neurotransmitter is going to diffuse and bind and cause ion channels to open on the postsynaptic receptor. The graded potential begins when we have our binding, and then the neurotransmitter is terminated. So we have degradation by enzymes, we have the reuptake by astrocytes, or the axon terminal itself, and then we have diffusion away from the synaptic cleft. This is the slowest part of the nerve impulse conduction, and it's actually called synaptic delay. It's because there's something that has to go on in order for that message to carry forward. So this really takes the longest. But by longest, you're talking about 0.3 to 5 milliseconds, right? So the postsynaptic potentials are graded with potential with strength determined by the amount into the neurotransmitter and the time the neurotransmitter hangs out in that area, right? So that is your steps for the chemical synapse. That's just kind of how we get that chemical sent from pre to post so that we can carry on with whatever the message was. So if we're thinking back to the steps of that muscular contraction, these are the details of how we get that message across the neuromuscular junction so that we can have that action potential in the sarcolemma. Okay, as I already mentioned, right, we've got this postsynaptic potential, these graded potentials with their strength determined by that amount of neurotransmitter and the time the neurotransmitter spends in the area. There are two types here. We have excitatory and inhibitory. Your excitatory postsynaptic potentials are going to move you toward action potential, so depolarization. They are graded, so they're not necessarily all or none. If you increase the neurotransmitter, you increase the excitatory postsynaptic potential. And stimulus will occur when threshold is reached, so summation. So we're talking about a graded potential. So we're going to look at those pictures in just a moment, but keep in mind, you have to summate your graded potentials to create an action potential. It could be EPSP, right? And it could be temporal versus spatial. So you've got to kind of take a look at that. So your stimulus here is what we're hoping for to have the action potential. And we're hoping to summate and reach threshold. Your inhibitory postsynaptic potential is actually moving away from action potential, so it's hyperpolarization. Based on the presence of neurotransmitters, we cause the movement of potassium out of the cell, therefore hyperpolarizing the cell and making it more difficult to attain the impulse. And again, we're going to look at comparison of action potential, graded potential in just a moment. Okay, these next few slides, we're going to make the comparison of the graded potential with the action potential. So we're going to go through the different parts of this. Some of this I've already mentioned. So the location of your graded potential is the cell body and dendrites, whereas the action potential is your axon hillock and your axon. Right? Your graded potentials are going to be short distance. Your action potentials are going to be long distance. Graded potentials can be various size and they decay with distance. Your action potential is always the same size. We're referring to all or none, and distance is irrelevant. Okay. To open the ion channels with a graded potential, that's neurotransmitter, could be sensory stimulus, light, pressure, temperature, but talking about a neurotransmitter, whereas the action potential is done by voltage or depolarization, which is triggered by that graded potential reaching threshold. The positive feedback cycle is used in action potential, and your action potential is voltage regulated, so you get that occurring when your sodium channels inactivate and the potassium channels open, whereas in a graded potential, your repolarization occurs when that stimulus is no longer present. For there to be a summation, you get a stimulus response that can summate to increase the amplitude of a graded potential, which does not occur in action potential because, again, action potential is all or none. You've got long distance signaling, and it's just that one nerve impulse with your action potential, where with a graded potential, you could get either an excitatory or an inhibitory possibility to either move toward or away from generating an action potential. This slide not necessarily as important as the previous three. This is just kind of summarizing your peak membrane potential. With a graded potential, they would have to add up to become an action potential, so you can move towards 0 and then towards 90. But you can see that peak of activity is quite small in comparison to reaching, say, 50, 30, plus 30 to 50 for your action potential. And then it just gives you a little bit of information about your chemically gated channels for graded potential versus voltage gated channels with action potential. There's more about this in your textbook. You can review it if you'd like, but the previous three slides from this one are the ones that have the kind of the meat of understanding the two side by side. All right, so our chemical connections between neurons, which are called our neurotransmitters. It's important to note that not all neurotransmitters excite the postsynaptic cell. We do have over 50 identified neurotransmitters. ACH was the first, makes sense. And we want to make sure that we know that it's determined by receptor type of the postsynaptic neuron. Okay? So the frequency of stimulation may cause release of various neurotransmitters. Not all are released every time. You can see a list here of some of our primary neurotransmitters. And those neurotransmitter effects may be excitatory or inhibitory, meaning that they could be depolarizing like glutamate, or they could be hyperpolarizing like GABA and glycine. This is determined by the receptor type. And for example, your ACH is the example of a excitatory at the neuromuscular junction, but it would also be inhibitory in cardiac, which makes sense a little bit if you think about it. And then we could be direct versus indirect neurotransmitter mechanism. If it's a direct mechanism, it's going to open ion channels and promote a rapid response, like what we see with ACH, but also with your amino acids. If it's indirect, it's going to act as a second messenger and promote longer lasting effects. These are going to be things like your catecholamines, indolamines, peptides, and dissolved gases. Your dissolved gases being things that impact learning and memory, which can be kind of a big deal. So your neurons function in groups with kind of a hierarchy as far as organization goes. Your billions of neurons in the central nervous system are organized into neuronal pools. They do that so that they can integrate incoming information and forward on any processed information. They could excite, facilitate, or inhibit. We have 4 different types of circuits that function. The first is diverging, so we have one input and many outputs, so it's going to amplify. We have converging, which has many inputs and one output, so it's going to concentrate. We have a reverberating or an oscillating circuit, where the signal is going to travel through a chain of neurons, each feeding back to the previous. Think here about your sleep-wake cycle or a repetitive movement such as walking. And then finally, we have a parallel after discharge circuit. This signal is going to stimulate neurons in a parallel array. So they might run in 3 parallel pathways that eventually converge. But because they potentially reach the output at different times, it may cause a burst of impulses called an after discharge. And last but not least, we have our neural processing. We have 2 ways that the processing of input information occurs. We have a serial processing where the input travels along one path to a specific destination. It's predictable, it's specific, it's all or none, and it's our reflexes. So they're rapid and automatic. This is that reflex arc with its 5 components: receptor, sensory neuron, CNS integration center, motor neuron, effector, and that's what you see here. 5 steps, quite predictable, one path straight through. Then we also have our parallel processing, which is where our inputs are segregated into multiple pathways. One stimulus might produce multiple responses, and information from each pathway is processed simultaneously. Right? You could get a higher mental function, or like a memory trigger, like a smell or a sound or a sight that's going to get multiple responses. And that's the end of part 2 and all the details that go with the nervous system. As far as taking your quiz on nervous tissue, should be good to go. There are a couple places I specifically said hint, hint. One of them is on this, happens to be on this last slide, so make sure you know the difference between these 2 types of neural processing patterns. And good luck. Let me know if you have any questions. Don't forget to check the announcement board.